Welcome, class, to the second half of part 3. We'll be going over part D in this lecture recording. You're still responsible for the information in part C, but that takes you through a good review of all the organelles inside the cell. So review part C on your own, and this lecture recording will be on part D, which talks about the cell cycle and protein synthesis. So there's two major periods of the cell cycle: interphase, where it takes on its normal metabolism activities and it grows, and then the cell division, or the mitotic phase. Interphase, again, is the period from cell formation to cell division, when the cell will carry out its routine activities and prepare for cell division. During interphase, the nuclear material will be in the uncondensed chromatin state, and interphase consists of subphases, which include the process of DNA replication. So in the subphases of interphase, G1 is the vigorous growth and metabolism, S is the synthetic or DNA replication phase, and then G2 is the preparation for division. And this is a good animation showing you the interphase stage and then the steps of the mitotic phase. You can see here how there's different checkpoints, G1 and G2, kind of as a good checkpoints for the cell that they have to get through before going on to the next phase. DNA replication is important for cellular division so that the cell, when it divides, can pass on a genetic blueprint to two identically genetic daughter cells. Prior to division, the cell will make a copy of its DNA, and it does that when the DNA helix unwinds and unzips. The replication fork is the main point where the strands separate, and the replication bubble is the active area of replication. So each strand basically acts as a template for a new complementary strand. RNA will start the replication by laying down short strands that act as a primer. DNA polymerase is the enzyme that attaches to the primer and begins adding nucleotides to form a new strand. So we have kind of this leading and lagging strand that are formed as the DNA unzips. And again, the end result is just replicating the DNA into two new identical daughter DNA molecules that are completely identical to the original. And then during mitotic cell division, one complete copy will be given to a new cell while one is retained in the original cell. So here's a look at replication of DNA, taking our DNA, unwinding it at this enzyme, and then each kind of unwound strand, the old strand goes through the DNA polymerase while new nucleotides are formed to create a new strand of DNA. Here's a great animation showing that. Cellular division then, we call the mitotic phase cellular division, mitosis and cytokinesis. Control of cellular division is crucial so that cells will always divide when necessary. So we're going to talk about the cellular cycle, the mitotic phase now. It's divided into 4. So mitosis is the division of the nucleus in which the duplicated DNA is distributed to 2 new daughter cells. There's 4 stages of mitosis to ensure that each cell receives a full copy of replicated DNA: prophase, metaphase, anaphase, and telophase. Prophase is divided into 2 parts. Early prophase, the chromatin will form invisible chromosomes, and each chromosome and its duplicate, called sister chromatids, are held together at the center by a centromere, and the centrosome and its duplicate will begin synthesizing microtubules that push each centrosome to opposite poles of the cell, and this is called the mitotic spindle. Late prophase is when the nuclear envelope starts to break up, and the microtubules will attach to specific areas on the centromere to try to pull the chromosomes to the center or the equator of the cell. So this is a look at interphase, normal cell growth, metabolic activities. This is a look at early prophase. You can start to see the mitotic spindle starting to form, which will again attach to the chromosomes. And then late prophase you start to see the mitotic spindle attached to the centromeres on the chromosomes, which will push them towards the center of the cell. Metaphase is then when the centromeres of the chromosomes are precisely aligned at the cell's equator on an imaginary plane called the metaphase plate. And metaphase is really fun to see under a microscope because it's really easy to spot the cell in the metaphase stage because all the chromosomes are lined up very nicely along this equatorial or middle of the cell. Anaphase is the shortest. This is just when the centromeres of the chromosomes are split apart at the same time. So each sister chromosome will now become a separate chromosome, and chromosomes are pulled toward their respective poles by motor proteins of kinetochores. And here's anaphase. You can see the daughter chromosomes are being pulled apart. Then telophase begins when the chromosome movement stops. Each set of chromosomes will uncoil to form chromatin, and new nuclear membranes will form around each chromatin mass as the nucleoli reappears and the spindle disappears. Cytokinesis is just then when the cells, the two daughter cells, will be pinched apart to become their own cells at this point. And we call this kind of area where they split is the cleavage furrow. So here's a picture of telophase and cytokinesis. You can see the cleavage furrow forming and then the nuclear envelope reforming around the new chromosomes. Great animation on mitosis. The cell division is easily controlled by go and stop signals, and these are just important to ensure that the cell divides when it's supposed to. Different chemicals can be a go signal, like a growth factor or a hormone, and stop signals can be if there's enough space for it, like contact inhibition. Two groups of proteins are crucial to the cell's ability to accomplish the S phase and enter mitosis, and those there. These are checked, and if some divide, the cell reboots itself. So again, to look at the cell cycle, we just went through interphase and mitosis, the mitotic phase. Protein synthesis then. So we're going to go over protein synthesis, the two videos that I posted in a recent announcement. It gives a great visual. Your DNA is a master blueprint that holds the code for protein synthesis, and DNA will direct the order of amino acids in a polypeptide because amino acids make up a protein. A segment of DNA that holds the code for one polypeptide is referred to as a gene, and the code is determined by the specific order of nitrogen bases: adenine, guanine, thymine, and cytosine, and they're always abbreviated A, G, T, or C. The code consists of 3 sequential bases called a triplet. So GGC will be a guanine cytosine nitrogenous base that codes for the amino acid proline, whereas GCC will code for arginine. Each triplet specifies the code for a particular amino acid. Genes are composed of exons and introns, where exons are part of gene that actually codes for the amino acids, and the intron are non-coding segments interspersed among exons. So here's a good look at DNA and showing how RNA is a little bit different from that too. The role of RNA, so RNA is the go-between molecule that links DNA to protein. So DNA gives kind of the genetic blueprint, and then RNA carries it out to actually create the protein. RNA will copy the DNA code inside the nucleus and carry it into the cytoplasm to ribosomes. All RNA is formed inside the nucleus, but RNA differs slightly from DNA, which that previous animation will show, in that uracil is a base that's substituted for thymine in RNA, and RNA has ribose instead of deoxyribose sugar. There's 3 types of RNA, whether it's messenger RNA, ribosomal RNA, or transfer RNA. Messenger RNA is single-stranded. The code from the DNA template strand will be with complementary base pairs, resulting in a single strand of mRNA. And this is a process referred to as transcription. mRNA will maintain the same triplet code or codon from the DNA. Ribosomal RNA is the structural component of ribosomes, the organelle inside the cell where protein synthesis occurs. And along with transfer RNA, will help to translate the message from mRNA into a polypeptide, which is just a growing chain of amino acids, which will make a protein. Transfer RNA is the carrier of the amino acids, so it has a special area that contains another specific triplet code, which we call the anticodon, that will allow each transfer RNA molecule to carry only one specific amino acid. The anticodon of the transfer RNA molecule will complementary base pair with a codon of an mRNA at the ribosome, where it will add its specific amino acid to the growing polypeptide chain. And that process is referred to as translation, when the tRNA brings the complementary base pair codon to the ribosome that will add its specific amino acid to the growing polypeptide chain. So protein synthesis occurs in two steps. Transcription is where the DNA information is coded into the messenger RNA. This happens inside the nucleus. And then the mRNA takes that information out of the nucleus and works into translation at the ribosomes. And when the mRNA is coded to assemble polypeptides inside ribosomes to create proteins. So here's a look. This is just a really simple representation, but I like this because it shows how DNA is transcribed to a single RNA, mRNA molecule. The mRNA molecule will then carry that genetic information out of the nucleus into the ribosomes, where translation occurs, where transfer RNA molecules will bring amino acids to create a growing polypeptide chain. And a polypeptide chain is just a chain of amino acids that will form a protein. Transcription, so here it kind of takes you through a little more detail of transcription, the process of transferring the code held in DNA gene base sequence to complementary base sequences of mRNA. You can read through the transcription factors there on your own. Transcription is broken into these three phases: initiation, which will separate the DNA strand, elongation, and then termination. Transcription will stop signal. So here's initiation, and I'll let you read through this on your own. Elongation, where we grow the new mRNA strand. Again, the mRNA is just the information taken off of the DNA, but in a single-stranded format. And then the termination, when it reaches the termination signal, the RNA polymerase and the complete mRNA transcript will be released to get the completed mRNA strand. The mRNA strand will then have to be processed, so introns will be removed by special protons, leaving only the exon coding regions, because the exon coding regions are what will actually code for the protein. This processing step is important, just kind of as a way to make sure that we're going to create the protein we want to, and that all the genes are in the correct place and there's nothing wrong or damaged. Translation is then the complex steps of protein synthesis, where the language of the nucleic acids, the base sequence, is translated into the language of proteins, the amino acid sequence. And the process involves the messenger RNA that came from the nucleus, the genetic code, the transfer RNA in ribosomes, the translating events, and sometimes the rough endoplasmic reticulum. So the genetic code, again, is each 3 base sequence on the DNA. It's represented by a complementary 3 base sequence on the mRNA called a codon. And there are 64 possible codons, 4 bases. We get a total of 4 kind of arrangements here, or 64 different codons. There are 3 stop codons, which will tell kind of the process to stop, but the rest of the codons are all for different amino acids. There are only 20 possible amino acids, so that just means that some of the amino acids are represented by more than one codon, and redundancy will help protect against a transcription error. So here's a look at the first base, whether that's adenine, guanine, uracil, or cytosine, the second base and the third base. So you can see all these different codons and how each kind of triplet will lead to this abbreviation for an amino acid. So different codons can code for the same amino acids. This is phenylalanine, leucine, iline, valine, alanine, threonine. So each of these abbreviations just stands for one of the 20 amino acids. The role of transfer RNA then. So transfer RNA binds a specific amino acid at one end. So that's And once the amino acid is loaded onto the transfer RNA, it changes its name, and the anticodon at the other end, called the head, is a triplet code that determines which amino acid will be bound at the stem. So for an example, the tRNA with the anticodon UAU will only be able to load a methionine amino acid to its stem region. The anticodon of the tRNA will bind only to the codon that's complementary. So for example, if the codon is AUA, only a transfer RNA molecule anticodon UAU will be bound, because adenine always complementary paired with uracil and opposite. Guanine is always complementary paired with cytosine. The ribosomes will coordinate the coupling of the mRNA and the tRNA, and the ribosomes will contain one binding site for the messenger RNA and 3 binding sites for the transfer RNA. So the sequence of events in translation, it'll occur in 3 phases. It requires energy or ATP, protein factors, and enzymes in initiation, elongation, and termination. Initiation is shown here, where the small ribosomal subunit will bind to the specific initiator transfer RNA. The anticodon of the initiator transfer RNA will bind to the start codon. The large ribosomal unit will then attach, and we get this initiation process to start. Elongation starts to occur while the transfer RNA will bind the complementary codon in the A site. The peptide bond will form so that we start to link amino acids together. And translocation just means that the ribosome will then shift down 3 bases of messenger RNA, displacing the transfer RNA. And then elongation, once the A site is empty, a new transfer RNA can enter. Only after a portion of mRNA is read, additional ribosomes may attach to already read part and start another round of translation. So this just shows the growing polypeptides coming out of the ribosomes. Here are the ribosomal subunits coming together at the start of the mRNA molecule. The polyribosomes are shown here, and then the completed polypeptide is formed and the ribosome subunits detach. Termination is then when one of 3 stop codons on the messenger RNA enters the A site, so the translation ends. So we know that that protein has been made. The final polypeptide product will be further processed by other cell structures into its 3D structure. So again, translation is the process by which genetic information carried by an mRNA molecule will be decoded in the ribosome to form a particular polypeptide, which is just a chain of amino acids, which will form a protein. These slides take you through a review of what we just talked about. And again, I would encourage you to watch the animations in these PowerPoints, as well as the YouTube videos that I posted on the most recent announcement. So again, feel free to pause the video at any time to read through these kind of abbreviation or summary of what we just talked about. All right. Here's the rough ER processing. So what this just talks about is the rough endoplasmic reticulum will go on and process those proteins a little bit more and then send them off in vesicles to where these proteins need to go, whether that's inside the cell or outside the cell. You don't need to know all the details about rough endoplasmic reticulum processing of proteins. I don't think I asked you any questions about that. So here's again a good summary from DNA to proteins. The complementary base pairing directs transfer of genetic information in DNA into amino acid sequence of proteins. DNA triplets are coded to mRNA codons, and mRNA codons are base paired with tRNA anticodons to ensure the correct amino acid sequence. So this shows the information transfer when we talk about codons and anticodons to give you a little more help here. So here's a DNA base sequence in triplets of the gene codes for the synthesis of a particular polypeptide chain. The mRNA base sequence of the transcribed RNA chain will complementary base pair. So you'll see that thymine will always pair with adenine and cytosine will base pair with guanine. The difference, again, between RNA and DNA is that adenine would normally pair with thymine in a DNA strand, but in the RNA, the thymine is replaced with a uracil. So whenever you see an adenine, that'll always complementary base pair with the uracil. The tRNA, again, is the transfer RNA molecules that just bring the correct amino acid to the spot. So here we have our mRNA. It's codon of the tRNA molecule also has to be complementary to the mRNA, and that will bring up the correct amino acid to make the polypeptide chain. And again, when we say polypeptide, that means many peptide bonds, because each amino acid is bound together by a peptide bond. Eventually, there's a start and a stop codon, which just tells the ribosome and when to detach. So it helps us know when to start the translation process and when to end it. DNA codes for types of microRNA and small interfering RNA. And you don't really need to know that. A couple of words that you should be familiar with need to be taken out of the system. So autophagy. All right, and I lost connection, so you saw me there for a second, but I wanted to talk finish this. So this is the self-eating process of disposing of non-functional organelles and sweeping up the cytoplasmic bits by forming autophagosomes, which can then be degraded by lysosomes. The proteasomes disassemble ubiquitin-tagged proteins, which recycle the amino acids and the ubiquitin. So just damage. Apoptosis is also known as programmed cell death, which causes certain cells to neatly self-destruct. This process begins with mitochondrial membranes, which leak chemicals that activate called caspases, which can cause degraded cell death, dead macrophage. Go ahead and watch the videos posted to try to help understand this a little bit better. And again, please email me or message me with any questions.